technology. So I'll pass now, Javi.
Thank you. Good evening, everyone. Can you hear me well? Excellent. Welcome and thank you for joining us tonight for a fascinating talk by Dr. Alex Kagan, who will navigate us through how nature's solutions could unlock secrets of longevity and disease resistance. Alex did his undergraduate in archaeology and anthropology here at the University of Cambridge. He then went to the Max Planck Institute of Evolutionary Anthropology in Germany, where he did his PhD with Dr. Svante Pääbo, the Neanderthal guy. Not that he is a Neanderthal, but yeah, maybe he has some Neanderthal genes there, but on the genomics of primates and animal domestication. At the end of his PhD, Alex became excited about the emerging field of somatic evolution, the evolution of the body. As scientists, we're realizing that the same evolutionary processes that occur within species also occur between cells in our bodies as we age. Then Alex came to the Sankar Institute here in Cambridge for a postdoctoral appointment, switching from studying evolution between species to study evolution within our own bodies. Alex was involved in developing methods to enable us to study these processes, which he will talk about tonight, and applying them to look at how different species age, including us. Alex started his own lab just under 2 years ago at the Department of Genetics in Cambridge, where they focus on studying the genomic aging across species with diverse lifespans and life histories. His work has profoundly impacted the pursuit of healthy aging and the possibilities of cancer resistance. Throughout his career, Alex has also enjoyed drawing and using illustrations to keep communities communicating science, some of which this art, these illustrations, we will see tonight through his talk. As an illustrator, geneticist, and explorer of animal DNA, Alex will offer a new perspective on this tapestry of life. So please join me in welcoming Dr. Alex Kagan for this special talk on the impossibility of whales. What can the genomes of extraordinary species teach us about human health?
Thank you very much. Thank you for the very kind introduction. And thank you all so much for being here this evening. It's really great to see so many people here. And I hope you'll find the next hour or so entertaining and informative. And I think hopefully most of you will have postcards on your seats. So there's a lot of, yeah, have a look. So there's a lot of interest in Cambridge about healthy aging research, and we'd like to invite you to give your thoughts on, what we should be focusing on. So feel free to leave comments, and we'll take that on board. Maybe you'll think we shouldn't waste our time with all of these weird species, and we should look at something else. But it'd be great to hear from you all. So good evening. So I'm going to be talking to you about our research looking at species with extraordinary lifespans and what we can learn from these species about hopefully living healthier lives. So let's get going. So you may know that the typical human lifespan can be represented as about 4,000 weeks. That's about 77 years. So each of these dots represents one of those weeks. It's a bit scary seeing them all up there like that. In dark blue are the number of weeks I've already used up, so that's extra scary for me. And it's already out of date, so it gets even worse. But we know that life isn't just about how long we live, it's about how well we live in those years. And we know that, thankfully, due to improvements in medication and sanitation, more of us are living longer than ever before. But we also know that as we get older, all those weeks aren't equal in terms of health, that we often have a decline in health as we age. And so in our latter years, our health typically deteriorates. We start to suffer from multiple morbidities, many different chronic ailments that lead to a decrease in health in the latter years of our lives. And this isn't just a personal issue, it's also a major global challenge. So we know, typically in human history, our populations have been dominated by young individuals who, the kind of social contract is we all work, and then we support the minority of elderly individuals. But due to all these improvements in healthcare, we're now living for longer than ever, and this is leading to profound changes in the demography of human populations. So we're now moving from demographic pyramids to more kite-like structures, and it's particularly advanced in countries like South Korea or Japan, where you have many more older individuals in the population due to living for longer and a declining birth rate. And this is really a demographic time bomb that's approaching many human nations. It's going to lead to problems like lower tax revenue, but there's going to be a need for higher healthcare spending, but fewer people in work. So this aging population is a major personal and social challenge. And there's no real simple single solution to this, but one thing that we think could help us as individuals and as populations is if we could increase the human health span. So not necessarily living longer, but could we live younger for longer? So could we avoid that decline in health towards the latter years of our life? And essentially the goal would be, can we die young as late as possible by improving our health so that we're all sprightly and young as we get older? But to achieve this, to improve the human health span, we really need to know what is aging and how can we extend healthy aging. And despite centuries of interest, we're still struggling to understand what actually causes us to age and what causes that decline in health. There's still debate about what aging even is, but we generally think of it as a deterioration in physiological function over time. So we get a decline in our function, our viability and resilience, and an increase in our vulnerability to disease and chronic conditions as we get older. We all recognize aging, but it's actually a very complex multifactorial process. There's lots of different changes. And we know that we don't all age in the same way at the same rate. Some of us age faster in certain aspects than others. But together, this leads to an overall decline in resilience as we grow older. And so as long as humans have been around, we've been thinking of how can we avoid this aging process, even though there's some positive parts as well. But the human history is full of these stories of quests against aging. So just to kind of give a bit of context for where we are today, the oldest surviving piece of literature, the Epic of Gilgamesh, revolves around Gilgamesh's quest for immortality. So that's our oldest written story. Then, later we've got the Arthurian legends of the Holy Grail promising immortality. And then, moving on a bit, we have this quest for the Fountain of Youth. And there were tales of Spanish conquistadors setting sail to Florida because they'd heard rumors that that's where the Fountain of Youth was. Though if you look at the demographics of Florida now, it's probably not the best place for where you'd expect a Fountain of Youth. But an important distinction is that apart from obviously being mythological stories, these are all quests for immortality, for living forever. And there are still scientists trying to achieve that, but what most scientists are focused on isn't making us live longer, but just making us live healthier for longer. So we still have the same lifespan, but can we fill that with healthier years? Although it's an interesting thing to debate, can you achieve one without the other? Can we increase the health span without increasing the lifespan? And that's something that we still debate. So kind of moving from the myth to the modern era in science, there's been a lot of research over the decades into healthy aging. A lot of this has been done in mouse models. And so, mice studies have made a huge contribution to our understanding of aging. Studies where we give mice different diets or pharmacological interventions and try and see, can we make them live longer or healthier for longer? There have been some great successes in this. So particularly things like dietary or caloric restriction has been really effective at extending mouse lifespan. So that's something we're interested in with humans. This idea of fasting or limiting dietary intake, could that extend lifespan? But although mouse studies continue to make a really important contribution, I think you can all probably imagine there might be some problems with using mice to study human aging. And one of the obvious ones is that mice don't live very long. So a 2-year-old mouse is an old mouse. And even if we can increase a mouse's life by 40%, which is what some of these interventions can do, they're still living less than 3 years. So it's still not a huge, compared to our lifespan, it's not a huge improvement. Of course, that can still be a helpful model, but I think there's probably a lot we could gain from not just looking at mice. And although there are benefits to these model organisms, we can keep them easily in the lab. There's all these species out there with remarkable lifespans, and what could we learn from looking at these kind of natural experts, species that have already achieved far longer lifespans than our own, that have been traditionally very difficult to study because we can't keep them in the lab. But as I'll talk about today, we now have new genomic methods that are enabling us to start to study the biology and the genetics of these really long-lived kind of super-aging species. So just to begin with, I'll take you on a little safari through some of the species that longevity researchers get really excited about before kind of diving into the biology of how we're now studying them. This is all the fun stuff. So even within mammals, there's a really large diversity in lifespan. There's the mouse that lives a couple of years. Humans are actually towards the kind of longer end of mammal lifespan, living about 70 to 80 years. But we've long suspected that some of these like majestic ocean giants, the whales, can live far longer than we can. But it's very difficult to know how long a whale lives. They don't travel around with a passport, so we kind of have to find ways to infer how old these species can live. There have been tales for some of these species, like the bowhead whale from natives from Canada and Greenland, who've lived with these species for many generations. describing how they know from recognizing individuals that they typically live 2 human lifespans. But we've been trying to quantify that, and recent findings have kind of suggested that that is indeed correct. And we can do this due to a kind of quite macabre way of dating whales. So many people know from the study, the novel of Moby Dick, that hunting whales used to be really big business because of their blubber. And there are many whales alive today that still carry in their blubber harpoons from failed whaling attempts. And many of these harpoons were just produced for very short periods of time because the technology was evolving. So by finding whales with certain harpoons in them, we can actually date when that whale must have been alive when it was hunted and survived. And there are particular harpoons that have been found in bowhead whales that were produced in the late 1800s. And we know they were normally only hunting adult whales because they were worth more. So based on this inference, we can infer that some of these bowhead whales are probably at least 200 to 250 years old. And what's really mind-blowing is that that means that there must be bowhead whales alive today that were swimming in the ocean before Moby Dick was even written. So it's kind of blows my mind. Oh, and also, I thought I should mention as we're here, some of these bowhead whales would have been alive before the Cambridge Union was even founded. So they've been around for a long time. So moving on from the oceans around Greenland to the plains of East Africa, there's another species that's really popular for aging researchers, but you wouldn't actually see it just walking around. You'd have to bring a shovel. But if you were to dig a little deeper, you might find a beautiful species called the naked mole rat. It's affectionately described as a cocktail sausage with teeth. There are worse ways to describe it. But everything about these animals kind of is unusual, and we think of them as kind of rock stars of the aging field. They're eusocial, so that's something we usually associate with ants or bees, which means there's a queen who breeds, and then they have a little army of workers who support them. So it's really rare for a mammal to have that. They're super long-lived, so they don't live as long as we do, but for a rodent, it's pretty exceptional. They can live regularly 20 to 30 years, compared to their relatives, the mice, that just live for a couple. And they're almost completely cancer-free, so thousands have now been studied, and you only see cancer in a handful of individuals, whereas the majority of lab mice tend to get cancer if you keep them alive long enough. And they're pocket-sized, so they're really amenable to lab studies. So in fact, the Department of Pharmacology in Cambridge has one of the few naked mole rat populations where they study them for different traits that make them really unique. So there's some really interesting kind of super ages within mammals. And then as we go to other vertebrate species like the reptiles, other vertebrate groups, we get species like the giant tortoise, very charismatic, slow-moving, regularly living 150 to 200 years, so far longer than a typical human lifespan. And then if we go to the extremes, we have, at least for the vertebrates, the Greenland shark. So this is thought to be the longest-lived vertebrate, and we think it can live 300 to 500 years. And so continuing on the literary theme, that would mean that some Greenland sharks were born before Shakespeare arrived on the scene. So they've been moving around in the ocean. How good they are as models of human aging, still debatable. They are cold-blooded. They kind of live in slow motion. They move at 2 kilometers an hour in the ocean. But nevertheless, they're a vertebrate that can potentially live for half a millennia. And then as we get further away from vertebrates, things start to get truly bizarre. You get species that are potentially immortal. So aging isn't even a concept to them. Whenever the species has immortal in the title, you can guess that longevity researchers want to study it. And so species like the immortal jellyfish, they have this really neat trick that when they get stressed, they can revert back to a juvenile form, a polyp, which is a bit like a butterfly turning back into a caterpillar and then waiting for conditions to improve. Or a bit like if you had a stressful week at work and you decided to become a toddler until things improved. So obviously, the further we get away from vertebrates, the harder it is to model. But studying these species with completely bizarre life histories really gives us a potential window to understand what are the real principles of healthy aging. And so we know species can have extraordinarily different lifespans around the globe, but we still don't really understand what causes this variation. Why does a bowhead whale get to live for 200 years, whereas we live for 80 and a mouse lives for 2 or 3 years? And again, it's kind of a famous quote that if we want to make sense of things in biology, nothing makes sense except in the light of evolution. And given that aging is a biological process, we can therefore apply the lens of evolution to understand animal aging and longevity. And when we do this, it starts to make sense of some of the trends that we see. So when we look at that curve of human health over time, we see that there's this decline in health later in life. And from an evolutionary perspective, this makes sense because natural selection, unfortunately for us, really just cares about how many offspring we have, what's our fitness in the next generation. And so if there are variants that arise in a population that make us unhealthy before we reach reproductive age, natural selection is very good at getting rid of them. But if the variants happen to make us sick later in life after we've already passed on our genes to the next generation, natural selection can't do much about that. It doesn't really care because it's not affecting how many children we have. And so we see within species that chronic illnesses tend to occur after peak reproductive age. And we call this selection's shadow, essentially that natural selection is very weak to variants that affect our health after reproductive age. They're just invisible to natural selection. But we see, nevertheless, there are species that have reproductive ages much later in life and much earlier in life. So why is that? It kind of just pushes the question further. Why do some species reproduce very early and have very short lives? And we can understand this through the lens of life history. So it's kind of intuitive to you, I'm sure, that some species kind of take this approach of live fast, die young, and some species take it much slower, grow slowly, live long. And we think we can explain a lot of this due to extrinsic mortality. So there are some species like the mouse where the risk of dying every day is very high. They're very small. They're very likely to be eaten by a predator. So for them, the best strategy to have a lot of offspring is to grow really quickly, have lots of kids, and just hope you have a lot of kids before you get eaten by an eagle or an owl. But if there's very low extrinsic mortality, then you're no longer in this rush. Species can start to grow larger, they can grow slower, and there are benefits to growing larger. You can compete better for resources, you can get more sex within your species, and so that's a good strategy if there's low extrinsic mortality. And in those species, there could therefore be a benefit to any variation that makes you live longer, because it takes time to grow large or to invest in your offspring. And so kind of depending on these external environmental factors, that can explain why we see some species with very long lifespans and some species with very short lifespans. And a really good example of this is in the rodents, we have the bats. So they're descended from ancestors on the ground who didn't live very long, but once they developed flight, they were therefore free from a lot of the predators on the ground. And then you get rodents evolving to live 30 to 40 years. So they've been freed from this pressure of external predation, and it can enable the evolution of longevity. So again, this tells us why we see this variation in nature, but it doesn't tell us the nuts and bolts of what's actually different in a bat from a mouse or a whale and a mouse that enables them to live much longer. And so this is where we try and understand what's going on underneath the hood within the organism. And still, it's very complicated. We still don't really understand what's going on. We know that there's many different hallmarks of aging. So there's all these different biological processes that change in our body as we age. But knowing which of these are cause and which are consequence of aging is very difficult. For example, we know that our teeth wear down as we age, but we wouldn't say that that's what's causing us to age. That's more of a consequence. And we still have that same struggle within the fundamental biology of aging, even at the molecular level. So I'm a geneticist, so obviously I'm biased towards thinking that the genetic changes are really crucial, but I think there's a good case to be made. Our genome really underpins how our bodies function and operate, and we know that we're starting to learn that our genome changes as we age, and that's what I'm going to talk about a bit today. So to tell you about how we're able to study what happens in our genome, in our cells as we age, I'm going to take you on a little journey through recent discoveries in human biology that hopefully will leave you with a slightly different understanding of what it means to age and what's going on within our body as we get older, and then motivates us to look at other species. So I'll start at the beginning. We all start life as a single cell, and then we rapidly develop into these multicellular miracles composed of trillions of cooperating cells. And we take all this for granted. We don't have to think too hard about it. Our cells just get on with this job of all working and cooperating together. And that's possible because all of our cells carry a copy of the rule book for how to behave in the form of our genome. About 3 billion base pairs wrapped around 23 pairs of chromosomes in the nucleus of all of our cells. And so you can think of this genome as containing the instructions telling our cells for how to behave. And if I was to hand you a book saying this book is essential to your life, keep it safe because if it gets destroyed, you'll die, you'd probably think, I need to keep that book very safe. I need to lock it away and not let anyone touch it. Unfortunately, our cells don't have that luxury because they don't have any memory. They have to constantly keep reading this book to understand how do I behave, what proteins should I produce at this moment in time. And as a result of that, the DNA in our cells is very vulnerable to damage. It's constantly being unraveled and read and wrapped around things. And so there are estimates that as a result of this, the DNA in every cell in our body is damaged about 70,000 times every single day. And we have 37 trillion cells, so that's a lot of damage. So that's potential disaster because these are the rule books for how our cells should behave. There's many different types of damage. You can have little single base pair breaks, or you can have the entire double helix snap in half. But fortunately, our cells are prepared for this damage. We have a variety of different repair systems that are looking for DNA damage, go in, try and repair it. The bad news is that these DNA repair systems don't all act with perfect levels of fidelity. So we have some DNA repair systems that are kind of like a master craftsman. They go in, they take their time, they beautifully repair the damage, so you don't even notice anything's happened and the DNA's left as it was before. But then we have other systems that just want to get the job done. They're in a rush, they'll botch the job, but they just want to fix the damage or move on. And this doesn't seem great. Why would evolution do this? But it's important because evolution just prioritizes good enough over perfection, which is a good life lesson as well. But you can see, just like this Wallace and Gromit cartoon or animation, if you imagine the train tracks like the DNA, there's proteins rushing over the DNA all the time. There's genes being expressed. And so if there's damage to the DNA, sometimes it's important that that damage just gets repaired as quickly as possible. It doesn't have to be a perfect job. You just have to get the train tracks clear. And so you have DNA repair systems that may incorrectly repair the DNA. They're just trying to get rid of the damage. And so over time, not all of this damage is going to be correctly repaired. Occasionally, typos will be introduced. We call these mutations, but you can just think of them as typos. They can be single letters. They can be whole chapters that get moved around. But over time, our cells are accumulating these mutations due to inefficient DNA repair. And so over time, because of the accumulation of this damage, our bodies become these molecular mosaics. You get mutations in single cells. Those cells divide. They pass on those mutations to the next cell. So we become these kind of patchwork mosaic clones. Fortunately, that's generally fine for our health. Most of the instructions in the genome, most of the text in the genome isn't really important. You can change it around. You can have typos. It's not in crucial areas. It's like having typos in the introduction to a cookbook. It won't really change the recipes. But if those mutations happen in key places within the genome, it can have dramatic effects on how our cells behave and cause them to kind of forget to be nice and cooperative and to kind of go rogue and act selfishly by themselves. And some of these changes we now know as the hallmarks of cancer. So you can have mutations that make your cells start to divide when they shouldn't, not switch off when they should. And we've known now for several decades that these mutations, we call them somatic mutations because they happen in our soma, our body, are our cause of cancer. So you have healthy cells, and some of them will get these mutations in kind of the important places in the genome, leading them to overproliferate. Over time, they accumulate more of these bad mutations, and that can eventually lead to the formation of a tumor, and ultimately it spreads or metastasizes around the body. And so traditionally, cancer genomics is focused on taking the cells from that tumor and sequencing them to try and identify the mutations that cause cancer. And that's been very successful. It's taught us a lot about the role that these mutations play in driving cancer evolution, but it's not really a complete picture. We've just focused on the end stage, and we've really neglected what about the other healthy tissue? What's going on in cells that are accumulating mutations, but they're not becoming cancers? What role could they be playing in aging and the function of our bodies as we age? And so we've wondered, are cancer and aging potentially two sides of the same coin, both driven by this process of an accumulation of somatic mutations with age? And one potential end stage of this process is the formation of a tumor and cancer, but could there be other processes in our body that are affected by these mutations beyond just cancer? So essentially, we have this idea of functional normal DNA. Proteins are produced normally, and we can continue to function normally. But with these mutations accumulating over time, they can potentially disrupt the shape of proteins or how proteins are expressed, and this could lead to problems in how our cells function. And you can imagine as we accumulate more and more of these mutations over time, it could be having an impact on our health. But the challenge is it's been very hard to study these processes in non-cancerous tissue because we're looking for mutations present in maybe just a single cell or small populations of cells. And we haven't really had the right tools to isolate individual cells or groups of cells and sequence them without having errors that just didn't enable us to study what was really going on in healthy tissues. So while I was at the Sanger, I was involved in developing a new pipeline, a newer method that would enable us to start studying what's going on in the DNA of the normal healthy cells within our body as we age. And to do this, we integrated kind of classic histology approaches. So we would take tissue biopsies, so very small, like a millimeter or two piece of tissue. We'd create microscopy slides, so these standard slides with very thin slices of tissue. And then the kind of magic to this method is something called laser capture microdissection. So this is where we take a standard microscope and you fire a laser beam through it. It's very fun. And you can microdissect out tiny groups of cells or even individual cells, isolate them, extract the DNA from those cells, and then sequence them. And so this is what that actually looks like. That's the laser beam firing through the lens of the microscope and then cutting out tiny bits of tissue. And then if we zoom in a bit further, this is what it actually looks like. So this is a video recording. You can see the laser cutting around the tissue. The tissue's on a plastic membrane on the glass slide. You put the slide upside down, and you hope when you cut around the tissue and the plastic will fall down into a little tube that you can collect and extract the DNA. But because these are so tiny, they look like a fleck of dust. Often with the static, they stick to the slide, so you have to widen the aperture of the laser and try and flick it off with the energy of the laser beam, and we call that laser catapulting. And if you're lucky, it'll fall into the collection tube, and if you're unlucky, it's like a tiny little sail, and it'll drift away on the wind, and you never see it again. So I spent a lot of my postdoc cutting a lot of these into tubes and never seeing them again. But so we did all these things to make sure there was no wind, anything to just minimize static, and eventually we got pretty good at capturing them. And so using this process, we could take tissue samples from different humans of different ages, from different cell types around the body, and we could start to map out what are the mutational processes going on within our cells as we age. And I think there were some pretty remarkable results. So we discovered that mutations are increasing linearly throughout our life, as we might expect. So for example, we could cut out these intestinal crypts, these little U-shapes that line our intestine and help us absorb food. And we could kind of count the number of mutations that are accumulating in these stem cells. And we see that we get about 50 mutations in every one of these stem cells every year. So is that a big or a small number? Kind of depends on your perspective. But given these cells divide about once a week, so it's about 50 divisions every year, a bit more, and they have to copy 3 billion base pairs of DNA. So it's essentially one error for every 3 billion base pairs it's copied, plus all other damage processes. So to me, that's really accurate. But nevertheless, because we live so long, over time this adds up. So by the time you're 40 years old, each of your intestinal stem cells already has 2,000 mutations. Yeah, and the number we've learned varies from cell type to cell type. So intestinal cells are on the high end. I didn't put skin here because it would be off the roof because of sun exposure, so they get loads of mutations from UV, so it's good to use sunscreen. But we also see consistent differences between other cell types. So even neurons that never divide get about 17 mutations per year. And again, is that a number that matters? Is that going to affect how our cells behave? Well, one way to think about it is that if you get 17 mutations a year in a 40-year-old, you get 680 mutations per neuron. We have 86 billion neurons, so by the time you're 40, that's about 59 trillion mutations across all the neurons in our brain. So from that sense, this could be problematic. But again, it depends. We're still trying to understand the effect that these mutations do or don't have on our health as we age, but we now have tools that enable us to study what's going on in the DNA as we get older. And interestingly, sperm and oocytes, our germ cells that get passed on to the next generation, have a much lower mutation rate than the rest of our cells. And again, we think this is thanks to evolution. Sadly, evolution doesn't really care about our soma. It's disposable. We just have to live long enough to reproduce. But evolution knows that mutations that accumulate in our germ cells could affect the health of our next generation. So it seems that we invest a lot extra in protecting those cells from mutations. And again, it's not just the mutations themselves. We also see that over time, just like in cancer, some of these mutations can affect how our cells divide. So over time, some of these mutations cause the cells to spread throughout our tissues, and we become these clonal mosaics, as I was mentioning, as we get older. And so these methods are revealing this kind of hitherto invisible landscape of clonal somatic evolution as we get older. Mutations occur, some of them cause the mutations to spread, and so you get little patchworks of cells that you can't see with the naked eye, but that all share the same mutation that's grown as we get older. And some of these are the earliest stages of cancer, before anything looks abnormal, but some of these may be having other effects on our health as we age. And so we've been learning about how these different somatic mutations may implicate our health. One example is in the blood system. So we have hematopoietic stem cells in our bone marrow that produce our blood. And again, we know that these cells accumulate mutations as we get older. Some of them, again, can give the cells an advantage. They start to dominate and take over the population. And we know that in individuals with higher rates of this, where we can detect these, we call them clonal expansions, these spreading mutant cells, you have a higher risk of cardiovascular disease, and you have a higher risk of overall mortality. So this is suggesting potential ways that these somatic mutational processes can have impacts on our health beyond cancer, although we're still trying to understand that connection between these processes and exactly how they're impacting our risk of these diseases. And kind of finally on this journey through the human body, we're learning that these processes don't just affect our own health, they can have transgenerational consequences. So we've known for a long time that paternal age can have an impact on the health of our children. So as men get older, the risk of having a child with several different developmental disorders can increase. One of these is achondroplasia or dwarfism. And it wasn't really clear why that effect increased so dramatically as we age. And it turns out it's because of these somatic mutational processes. So within the testes, you have spermatogonial stem cells that produce the sperm. Some of these can accumulate mutations that make them divide more rapidly. So as you age, they colonize the testes. And as a result, more and more of the sperm in the testes will contain these mutations that benefit the stem cell. But if those sperm then fertilize the egg, they'll have that same mutation that disrupts development, and that can lead to developmental disorders. So we're learning that these somatic processes affect many aspects of our aging process and even our reproductive health as we age. And I kind of like to think of this new way of looking at of mutation and evolution, not just between individuals, but within our bodies as we age, is really changing the way we think about ourselves. And it reminds me of these pointillist paintings where from far out, it looks like we're all individuals, and we're all taught at school that we have one genome that we inherit from our parents. But actually, when we look a bit closer with these new tools, we realize that we're full of diversity. And in fact, no two cells in our body share the same genome. Even from the moment we're born, they're already accumulating different mutations. And the older we get, the more variation there is because that process continues to accumulate. And this could have really profound impacts on us and our health. But the more we've learned about the humans, that just raises more questions and paradoxes about what's going on in other species. Because in fact, based on everything we've learned about human aging, it should be impossible for species like these large whales to even exist. Because in terms of cancer risk, we know that the more cells you have, the greater your risk of cancer. Basically, every cell has a small chance of turning into a cancer cell. And we know within a species like humans, the larger you are, you have a slightly higher risk of cancer. And for example, across dog breeds, we know that larger breeds have much higher risk of cancer and shorter lifespans than smaller breeds. And yet, if every cell within a whale had the same risk of cancer as a human, they shouldn't be able to reach even 1 year of age without being riddled with tumors if we modeled that out. But not only are they living, getting to adulthood and much larger than us, they're often living far longer than we do. And this has been noticed for a while. It's called Peto's paradox after Richard Peto, the epidemiologist who discovered it. And it's basically this observation that we expect larger species to have much higher rates of cancer because they have more cells. But they don't, not only do they not have higher rates of cancer, often they actually have lower rates of cancer. So species like elephants, we think, only have a much lower rate of cancer than we do. And just to put that in perspective, and from one of Richard Peto's papers, he mentions that you can essentially fit 10 million mice into a whale. I wouldn't suggest that you try doing that, but if you look at the body size, you can. And this really suggests that essentially for every kind of mouse-sized piece of whale tissue, that whale tissue must be about 10 million times more cancer resistant than mouse tissue. In fact, it's even more than that because whales aren't just bigger than mice, they live a lot longer. And so what this suggests is that large-bodied, long-lived species have essentially potentially solved the problem of cancer, or they're much better at resisting cancer than we have. And they probably have had to do this because if you want to evolve a really large body size, you have to overcome this problem of how do you get big and live a long time without developing cancer. And so I think there's a lot of value in studying these large, long-lived species and trying to figure out their secrets rather than just taking the approach of trying to make a mouse live longer. Why don't we look at what evolution's already come up with in some of these species? And so to do that, we first there wasn't really any data to look at this, but we were curious, in these large, longer-lived species, or species with a different lifespan, we know that mutations lead to cancer. They may also contribute to other aspects of aging. So does that mean that species that live longer are better at protecting their DNA and their cells than we are? And what could we learn from looking at this process? So we just wanted to generate some basic data on what is the rate at which different species accumulate mutations. Do species with longer lifespans accumulate mutations at a slower rate? Is their clock ticking at a slower rate? And is a mouse ticking at a faster rate if we look within their cells? So to do this, we collaborate with a lot of different groups, particularly London Zoo. We need to know how old animals are when the time they die so we can figure out how old are they, how fast are they accumulating mutations. So zoos are quite good for that because they have very good records on the age of the animals. We also work with different groups who specialize in working with different species. And we tried to get a wide diversity of animals for this first study. So we were very happy to have species like the naked mole rat, these very unusually long-lived species, large-bodied species like the giraffe. But there are still other, even bigger species we're currently in the process of collecting samples from. And the process is essentially we wait for the animals to die of natural causes. So we didn't harm any animals for this study. We just wait for animals to die naturally. And then during the necropsy report, they would collect a small bit of tissue for us, send that to the Sanger Institute. We would make the slides, and we could do our laser capture microscopy. And so, for example, this is the intestinal epithelium of an African lion from London Zoo. And you can see these are all individual crypts. And much like the laser pointer, you could actually use the microscope laser to cut out individual crypts, sequence the DNA, and then we could compare how many mutations are in a 5-year-old lion, for example, compared to a 5-year-old human. Are they accumulating mutations at a different rate? What's going on in their body? And so this is what we did with this study. We collected the cells from many different species, individuals at different ages within every species, and we were particularly focused on individual somatic mutations. So we weren't looking at, there's other types of variation you can get. You can get really large structural variants where whole parts of the genome move around. But for this first study, we're just looking at the kind of single letter typos within the genome. And when we did this, we found something really remarkable, which is that there is this really strong inverse correlation between species lifespan and mutation rate. So I mentioned earlier that we get about 40 to 50 mutations every year in our intestinal cells. When we look at the intestinal cells of other species, it's a very different story. So, dog has quite a short lifespan, gets about 300 mutations. And then if we look at the shortest lived species in our study, mouse, they get about 800 mutations every year in their stem cells. So that suggests that indeed shorter lived species aren't investing that same amount in DNA repair. And when we know how long all these species live for, so we can say, okay, a mouse gets 800 mutations a year and it lives for 3 years. A human gets 50 mutations and lives for 70, 80 years. How many mutations are all these species getting by the end of their lifespan if they were to live out a full life? And then it looks much more similar. It looks like all of the mammals we looked at are getting about 3,000 to 4,000 mutations by the end of their lifespan. And so what this is suggesting to us is that there's an evolutionary constraint here on somatic mutation rate, that indeed longer-lived species, there's some kind of selective pressure to reduce the rate they get mutations. And that's consistent with this idea that there could be a causal role for these mutations in the aging process. Although this is still correlative, so we want to do more studies to understand if these are really driving parts of the aging process. But again, so we see that there's this constraint here, suggests that it's important for longer-lived species to protect their DNA, but it doesn't tell us exactly how they're doing it. So there's currently studies from other groups that are shedding some light on this. So I'll talk through some of the different ways we think that can explain this observation we've made, that there's differences in mutation rate between species with different lifespans. So one potential solution you could imagine is just have more DNA repair proteins in the cell. So if there is damage, there's more repair kits around to find it. And there have been studies looking at gene expression in the cells of naked mole rats compared to mice and other species. And we see that in the longer-lived species, it looks like they just have more expression of these DNA repair kits. So this seems like one potential solution, is just to have more DNA repair around in the cell so that it can find that damage when it occurs. But another potential solution isn't to have more DNA repair kits, but to have better tools. You don't need more wrenches, you just need a better wrench. And there's a study just from last year in the bowhead whale, we think the longest-lived mammal, where it seems that there are slight changes in the protein structure of one of their DNA repair genes, specifically one that repairs these really serious double-strand breaks when the whole DNA double helix snaps. And it looks like when you take bowhead whale cells and human cells and you expose them to DNA damage that causes these double-strand breaks, bowhead whale cells are much better at finding and repairing this damage because they have this slightly different structure to one of their repair proteins. So it seems like there may be different solutions between different species in the approach that they take to healthy aging and avoiding these somatic mutations. But interestingly, one thing we found was that we saw this really strong correlation between lifespan and somatic mutation rate, but we didn't see an additional effect of body size. So if the whole solution to avoiding cancer was just having a lower somatic mutation rate, we would expect there to be an additional effect of not just living a long time, but having more cells. But actually, we see that this correlation with mutation rate maps really well to lifespan, but there doesn't seem to be a relationship with body size. And so that suggests that when it comes to this Peto's paradox of avoiding cancer, that there may be other solutions that have been found by nature. And we're still trying to understand what those are, but there's been some really exciting studies on elephant that suggest what one of these mechanisms might be. And it's slightly different to this idea of just repairing DNA damage. And so we can understand this if we know that there's a gene in our bodies called TP53, and it's really a kind of a guardian of our genome or a kind of policeman in the cell. So one of its jobs is if there's too much DNA damage in our cell, it tells the cell, okay, that's it, you've taken too much damage, you're too high at risk of becoming a cancer cell, so it's time for you to die or commit apoptosis. And so it's kind of a selfless act of the cell to kill itself so that we don't develop cancer. It's a really common event in humans. If both of those copies, we have 2 copies of this gene, one from our mother, one from our father. And if both are knocked out by mutation, that's a really common event for cancer because then that cell no longer has the brakes on. It can just keep accumulating DNA damage and become a cancer. And there are some individuals born with, we call it Li-Fraumeni syndrome, so they're born with just 1 copy of this gene functional. And they have much higher rates of cancer because they just need 1 mutation and they've lost all of their functioning copies of this kind of guardian of the cell. So people have looked at the elephant genome and they've realized that elephants have 40 copies of this gene. And and this could be, it still waits to be seen, but this could be one of the ways that these large-bodied species are able to resist cancer, is their cells are much more sensitive to DNA damage. So as well as potentially having a lower rate of mutations, if there are too many mutations, they're also better at getting rid of those cells. And so you might think, okay, we've solved it. We can just put extra copies of TP53 into our genome and we'll be fine. But unfortunately, it doesn't seem that simple. It rarely is. People have made mice where you put in extra copies of TP53. And indeed, it seems they get less cancer, but they also die much earlier, and it seems that's because they run out of stem cells, because now they're so sensitive to DNA damage that they just get rid of all of their cells and they have nothing to replenish them. So we're starting to understand potential mechanisms of avoiding cancer, but we still have a long way to go before we can take what we're learning from these species and apply them to our own biology. But what's really exciting about you could see if it's a negative or a positive result, but people looked at, of course, other large-lived species and thought, are there the same copies of T53 in whales or other large species? And it turns out it seems a unique solution to elephants. We don't see this in other long-lived species. But in a way, that's really exciting because we know that large body size has evolved independently at least 11 times in mammals and many other times across the tree of life. So this suggests that nature and evolution has found many different solutions to cancer resistance in these species, and they're just out there waiting for us to figure out what they are. And so now that we have the genomic tools, we hope that we can start to look at these species and understand their different mechanisms of cancer resistance and healthy aging. And so currently in Cambridge, we're in the process of starting to study the somatic evolutionary processes in these species. We've had to bring in a few new collaborators to do this, and so one of the key collaborators is the Cetacean Stranding Investigation Program, essentially CSI for whales. So we live on an island, we have a lot of coastline, and unfortunately, very often you get whales or dolphins washing up on the shore. And when that happens, someone will call, a dog walker will call it in, and there's this guy, Rob DeVille, who his job is to go out in a van from London Zoo or the Natural History Museum and do a necropsy on these animals to try and understand the cause of death. So they're doing research to try and understand why do mass stranding events occur, what's the role of things like submarine noise pollution, environmental pollution. But we can benefit from these studies to gain access to tissue that's very hard, as you can imagine, to access, to start to look at the somatic mutational processes in these absolute giants, to understand what's going on in their cells and how are they protecting themselves from DNA damage. And again, as I mentioned earlier, one of the really key species is the Greenland shark. We don't often find them in the UK, unsurprisingly. They're mostly around Greenland. But a few years ago, one of them made a really long journey and washed up in Cornwall. So we're currently at the Sanger Institute laser microdissecting out different tissues from this specimen and other Greenland sharks from collaborators with other groups in Denmark and Greenland to try and understand what's going on in these real superstar aging species in terms of their genetic diversity. And so the goal really is to seek inspiration from across the tree of life to complement the existing laboratory model organism studies. We're starting to collect samples from species with extreme differences in lifespan. As I mentioned, the Greenland shark. There's also species of really short lifespan as comparison. So there's a species of fish called the killifish that only lives for 3 months every year. By 2 months age, it's already kind of getting cataract formation in its eyes. So we can look at the differences between very short and very long-lived species. And there are species like the ant, where you have a species with the same genome, but the queen can live for 30 years and the workers are only living for a year. So what's going on in those species to explain those differences? And so there's many different ways to look at these challenges. I focus on the genetics, but there's many other aspects of aging, and we hope this is just one piece of the puzzle. And we're still far, unfortunately, I can't say here's a pill we've made from the elephant that we can give you to live longer, but I think there's a lot of mileage in this kind of approach, and a kind of metaphor I like for this is thinking of the Wright brothers who realized we can't fly, we don't have wings, but they sought inspiration from nature, from looking at birds and the mechanics of flight, and that led to the development of the airplane. And in a similar way, I think by taking inspiration from the natural diversity that's already out there, now that we have better tools for studying this genetic diversity in our bodies, we can start to now look at species like the elephant, species with extraordinary lifespans or extraordinary metabolisms. So I haven't even had a chance to talk about birds, but they're pretty extraordinary in terms of their cancer resistance as well. And this wasn't really available to us before because we just didn't have the right genetic tools. You need a kind of map when you study the genome of a species. You need a reference genome. So it's a bit like having the guide to a jigsaw puzzle when you're putting the pieces back together. And we've had mouse and human for a long time, but it's only in the last few years with projects like the Darwin Tree of Life at the Sanger Institute and others that we now have the kind of maps to start to explore the genomes of all these diverse species. And so it just gives us a much wider toolkit to start to study these. We have much more advanced methods for single-cell sequencing and other approaches we can use. And people even starting to culture cell lines in the lab, as I mentioned, of species like the blue whale. So we just have a much wider toolkit to study what's going on in these species. And so kind of to wrap up, I guess, I don't really, I talked about all those myths earlier, but maybe I was wrong. Perhaps the fountain of youth is out there. We've just been looking for it in the wrong places. And it was actually been all around us all along in the species that we share the planet with. And unfortunately, many of these large, long-lived species are critically endangered because of human activity, overhunting, pollution of the environment. So it's just another really important reason, if only for selfish reasons, to preserve these species, because they may contain within their genomes the secrets to helping us solve our challenges of living longer and healthier lives. So with that, I'd like to thank you all for your attention. I'd like to thank all the people who've been involved in this study, and I'd be really happy to take any questions and continue the conversation. So thank you.
Well, Alex, thank you so much for such an enlightening talk about a topic that is incredibly fascinating for biologists and non-biologists, for everyone who is interested in life and aging and lifelong, like quality. I think there's just so many questions, but I would love to, before I asking some questions, to open the microphone to the public, because I believe many of us have some questions. Is there a microphone we would pass around? So I think we already have a few questions over there. Yes, and there is one over there as well.
Could you say a bit more about the reproduction? Because the whale have only one baby. I think probably they only have one every 3 years, every 5 years. And then, as you said, that production is sort of the end of your life. So it means that if they have one less offspring, they live longer. Is that sort of?
I see this idea of why some species, how the reproduction leads to life longevity. So I think in species like the whale, they've managed to avoid this, by growing very large, they're escaping this challenge of extrinsic predation. So they don't have to worry about this high chance of mortality every day, mostly. And so this can then select for, we think, these longer lifespans where you have more parental care. And interestingly, this idea of parental care is one of the ways you can kind of escape selection shadow. So if living for a longer period does improve the health of your offspring, if you don't just forget about your offspring once they're born, but you invest a lot in looking after them, then there can be selection, we think, to extend lifespan. And so this is kind of called the grandmother hypothesis. And we think in some species like whales, potentially elephants or chimpanzees, there may be some evidence that having post-reproductive individuals around longer with all of their knowledge, especially these kind of complex social species, can provide a benefit to the offspring, children and grandchildren. And so that seems like one way, it's kind of a nice way to think about that, there's a use for us after we finish reproducing if we're helping our family out. So that's kind of one way that these species can still have selection for long lifespan, even though they're not producing large numbers of offspring.
That's a brilliant question and good point as well. There's another question over there.
Hi, I've got a two-pronged question. The first is, do you think that the solution to your study would come from the DNA of the animals themselves, or would it be like a mechanism you could apply from looking at their DNA that we could apply separately to human DNA? And if it was the case of the first outcome, how would you prevent that knowledge being used for the wrong reasons? Because whales have been hunted to near extinction for lesser treasures than an immortal lifespan.
Yeah, that's a very good point. So I'm hoping that this research wouldn't lead to any outcome where you would need to harvest the animal to get the treatment, like an ointment or something like this. So fortunately, it doesn't seem that is the case. We think, our goal is we'll recognize mechanisms of DNA repair, like, for example, this molecule, this better DNA repair protein, the bowhead whale. It wouldn't really make sense to try and harvest them and take it from them. We hope there's ways you could just synthesize either a similar compound and try and treat it for individuals. Then again, as I want to say, we're still at the early days of understanding, so we're far away from being able to just copy a DNA repair protein, deliver it to our cells. But we're hoping we can do develop some kind of drug or biomimicry rather than something that you could only produce. Because a kind of an advantage is these proteins are very similar to proteins in our own body. They're mammals as well. So it's really just single amino acid changes. So it wouldn't be beyond the realms of possibility to manufacture or alter proteins from another mammalian species and put those in. But we still have all the classic challenges of how would you deliver that to all the cells throughout the body continuously without problems. So yeah, that's a good point though. Yeah, I hope that it would never lead to needing to harvest the animals that we learn from.
I was very interested in the difference in the age of the ant and the queen ant, because honeybees feed the queen on a different diet, so royal jelly, and that's why presumably she lives longer. So is there a dietary influence and epigenetic effect?
Yeah, that's a really good question. So I'm not an expert on invertebrate aging, but from my understanding, yeah, it's clearly not a pure difference in the genome. They share the same genome. We think there can be epigenetic changes that can then trigger, for example, potentially differences in maintenance of the individual. But that's a really good point about the diet as well. So I didn't really even talk about metabolism, but we do think that, you know, the queens in these species that live a very long time, they're kind of outsourcing all the metabolic work of producing this food and kind of given a very high energy diet. So in a way you can think of it as a kind of, some people think of it as a distributed organism, and the queen benefits from all the workers generating the nice food and doing the work, and so they have much lower metabolic costs. And we can see when we look across vertebrates, you know, species that have lower metabolic rates tend to live longer. And like, you know, cold-blooded species like reptiles have much longer lifespans generally than mammals because they don't have to pay that energetic cost in the kind of reactive oxygen species that can be produced that are harmful to the cells. So yeah, I think that could definitely be an important part of that.
I would like to follow up on that question before we handle the microphone about the temperature. So I would imagine that the metabolism of the organism, how fast we process energy and food, etc., will dictate to some extent how long or short our clocks tick. Mm-hmm. So in a planet where we have a latitudinal gradient, an altitudinal, so temperature is not homogeneous across the planet. We have the poles, we have the equator, the tropics, and we have the altitude high mountains. For a given species, let's say humans or a white-tailed deer or something, a species with wide distribution, what would be the role that you think, or what do we know about the role of temperature in aging and life and quality of aging?
Yeah, that's a really good question. So I don't think it's a coincidence that a lot of those long-lived species, the bowhead whale, the Greenland shark, they're found at the extremes where it tends to be colder. And so you can kind of separate chronological age from biological age or metabolic age. And so, you know, the Greenland shark is living a lot longer, but it's kind of living in slow motion, essentially metabolically. And so the temperature can kind of have that effect, potentially, depending on the species and its biology, but certainly kind of like living in a fridge. Everything happens a bit slower, so you're living longer, but metabolically at a slower rate. And we think that similar things may happen with things like calorie restriction, that you're slowing down your metabolism. So that's a way to kind of chronologically extend your life.
Wonderful, thank you. We have a question over here, right?
Can you change how long you live by eating different proteins?
Yeah, it's a good question. So there's lots of different research into which diets are better for us for our longevity. I'm not sure of any specific protein. I mean, we certainly need proteins to be healthy, so you don't want to be deficient in any. There may be some studies I'm not aware of about particular proteins that increase longevity, but we do know that kind of not eating too much seems to be very good for longevity. So people who restrict and only eat when you're hungry and not binge eating can be very good for our health. But it's a very good question, and the role of diet in longevity is something that's of really great interest.
Just a note, this is from a talk from the Cambridge Philosophical Society last year. For all I know, you were in the audience. It was a fascinating talk from Jan Hogemakers, who's done the ultra short-lived mouse work, which may solve the other half of the problem. How do you turn the linear mutation rate into the classic picture of aging with wildly different effects in different tissues? And he's been studying transcription stalling in senescent cells. And there are a variety of types of DNA lesion in which RNA pol will just stop and not decouple until the whole genome is replicated. And they've observed that there are thousands, dozens to thousands of RNA pols stalled behind single ones often. The effect of this is that the longer the gene, the less it is expressed in increasingly senescent cells. And if you put the picture of this together, it has almost exactly the same picture as you see in aging tissues. And this may be the other half of the picture.
Yeah.
In any case, it's a fascinating bunch of papers to read.
Yes, yeah. And that is very interesting. Yeah, so I kind of talked about mutation, but there's another whole aspect to that. It's just the DNA damage itself. So even if the damage isn't repaired, that's things that are, you've got broken bits of DNA or lesions, things that stuck to the genome, and those in themselves can cause problems even without the mutations themselves. But it's very difficult experimentally to disentangle what's the role of the damage versus the mutation? And we're very good at measuring mutations now because we can sequence the genome, but it's a lot harder to kind of get the same scale of information on the damage itself on the molecule.
It seems to scale with the actual length of the gene before the introns are removed, which would map to it being just down to transcription story.
Yeah, that makes sense.
This might seem like an odd question, but I like the animals, and particularly hedgehogs and things like that. Is there any sort of correlation between the animals that hibernate and the effects on the aging process? So when they effectively sort of slow down and shut down, does that enable the animal to sort of repair some of the damage that's occurring?
Yeah, that's a really good question. So we want to start looking at some of these hibernating species to see, do they have a much lower mutation rate, or does that mutation rate lessen when they enter that kind of hibernation or torpor state? So we think it does. I mean, they're essentially shutting down a lot of the metabolic systems. There's also really other, beyond just mutation, there's fascinating bits of hibernation biology. These species like the bears that sleep for months, and then they can just get up within like an hour and their muscle mass is still there. So that would be a huge benefit for people who have bed rest and loss. We get really quick deterioration. Yeah, so there's groups studying that. They haven't cracked it yet, but if you could find whatever it is, that it would be great.
Well, I just to say that you explained it so well, because I'm not a scientific person, I struggle with lots of facts and stuff like that, but the way you explained it made it able for me to understand most of what you're saying.
Oh, thanks very much. I'm sure it was my fault the bits still were. Yeah, you're welcome.
How do you find out whether a whale has like cancer? Because obviously it's quite large, you can't really put it in like, MRI.
Yeah, that's a really good question. Yeah, unless you're like Damien Hirst. So I didn't touch on that, but yeah, we think that there are different rates of cancer in different species, but how do we even know that? It's really challenging, and there's such a huge bias in the amount of depth the pathologist goes to looking for evidence of cancer, depending on the species or the circumstance. Almost all of our data on cancer rates in animals comes from zoo studies, so we don't really have a good handle on what's going on in the wild. There are groups now from Arizona who are building portable MRI scanners to try and go out and give an elephant an MRI. You can imagine it's they're still trying to find a large enough shipping container to get it out there. But yeah, that's a really big challenge. It is noticeable though, when the whales do wash up, from my understanding from the cetacean team, is that they hardly ever, they do look occasionally, they look for cancer. It's a huge whale, so you can imagine they can't look everywhere, but they're not seeing evidence of, we might suspect small benign masses. It's very rare for them to ever see anything like that in the cetaceans that they do look at. But you're absolutely right, we don't have great data on, like we do in humans, for cancer prevalence. And obviously we only get the whales that wash up. Most whales never make it to the shore.
Well, I'll also follow up on that question a little bit and tackling on some of the other questions before. I would have imagined as well that, so if that the DNA duplication and reparation, and we do like all these Xeroxing photocopying, like grabbing a book with pages, and the more we get a copy, and then we copy the copy, it's just a matter of time until we start making mistakes. We flip a copy, we copy the same page twice, or we delete a word or something. So the more cells we have, the more printers we have that are potentially making errors. But then we have things like the whales that are so longevous. They have, I would imagine, more cells and bigger cells than a mouse. So it's like the paradox of the counterintuitive, how big animals such as a whale can actually live longer, even though they are more prone, because they live longer and have more cells, to get more of these copy errors. So how do we reconcile this?
Yeah, that's the key paradox that really motivates a lot of this research. And so essentially, when you think about it, we can take advantage of that all of these large species, they've had to overcome this huge challenge because of this paradox. So all these large-bodied species that have evolved independently are kind of like natural cancer prevention trials that evolution's been running for millions of years. And if we can figure out their solutions, the question is then, can we hopefully develop treatments that are based on those evolutionary principles, hopefully without having to harvest the animals and collect them, but take advantage of the designs that evolution's come up with and discover them. And we have hints from the elephant. Maybe you can have more copies of these tumor suppressor genes, but that's just one species, and we just have no idea for most of these species what's going on. So we're just at the beginning of this journey of being able to then interrogate what those mechanisms are, and can we use that to improve we don't want to have to wait millions of years until we get longer lifespans.
Right.
Can we learn from what's already out there in nature?
So I guess it's really exciting to know that we are at the beginning, the dawn of exploring this type of questions.
I think so, yeah.
Marvelous. And we have more questions over here. Yeah. Thank you.
Any practical advice for living longer?
I knew this question would come, so I should have prepared a better answer. I can give the normal spiel that we're just at the beginning of the journey, but based on what we see from the animal, no, I think from this research, no, but the fact that a lot of these species live in cold environments, I guess, go to Greenland. Yeah, exactly, while it's still cold.
Yes. We have, brilliant, we have one here. And then we have us also up here.
I guess it follows on from the last question. There was a Netflix series about blue zones, and the one that stood out was like the Sardinia one, and there's I don't know if it was a Florida one, but it seemed like warm countries with the blue zones where humans were living longer. Like, is that a piece of the puzzle that comes into it as well, or is that?
Yeah, it's a good question. There's no simple, clear correlation. Like when I say go somewhere cold, it's not that simple. There's all these other factors. There's obviously studying human genetic diversity, not just genetics, but places that live longer is another extremely powerful approach. You don't even have to translate what you find to another organism. That's just in our own species. So that definitely makes sense to look at those. There's a lot of debate recently that's been going on about those blue zones, that often they're regions where you may have high rates of tax evasion or people pretending they're living longer. So there's still debate about how many of the blue zones are really blue and how many are just people trying to collect pensions that they shouldn't be. So that's a challenge. At least the animals don't fill in forms incorrectly. So that's a problem we have. But yeah, that's a challenge we have.
We have a few over here, one and also the back, and here, one.
Two more questions? Okay, so two more.
Hi, I was just wondering how much variation there is in humans. You said the average was 50 bases per cell.
I feel like the mutations how much variation there is within humans and how quickly they get mutations.
Yeah, and so presumably is there a lot of variation, and those who get less mutations live longer?
That's a really good question. So I don't think we haven't had enough data to really test that yet, but there are groups looking at these mutation rates in like centenarians or people who live longer to see if there are differences. And we haven't measured directly the number of mutations, but there is some small evidence from centenarians that some of them maybe have differences in some of their DNA repair proteins. So we might expect that maybe they would have a lower mutation rate, but we've only just recently been able to actually measure these processes in humans. And we're still also looking at differences between the sexes as well, because we know there's differences between sexes in lifespan. Women tend to live a few years longer. And so far, we haven't seen a difference in mutation rate between the sexes. So this is just one piece of the story, and aging is more complex than just the mutations. And so far, we haven't seen differences in mutation so far that would explain differences between individuals or between the sexes. It's more so far at the species level. But as we get more and more data from humans, we should have more power to try and see if there are differences either between certain cell types within the body, because we know every cell type has a slightly different mutation rate. And so maybe some of those are more important. And that could help us figure out, all of our cells get mutations, but which ones are really critical for healthy aging? Does it matter if your esophagus gets mutations compared to the cells in your neurons? So we're still trying to understand that relationship.
Taking on that one very quickly, I guess a mutation is not a bad thing per se. A mutation can be good, can be neutral, so it doesn't have a yay or nay, or could be deleterious, right? So it's not just also only about the number of mutations, but where they are occurring and what type of mutations.
Yeah, that's a great point. I just talked about mutations as a bad thing, but there are cases, for example, if you have, there's some recent evidence, if you have certain liver diseases, you can get somatic mutations that kind of rescue the tissue. So you get a mutation that kind of compensates for the issue. If you're born with hereditary diseases, you can get we call it somatic rescue. So those mutations can actually come and restore function to tissues that aren't functioning well. It doesn't often happen because the mutations, whether they spread, just depends if it's good for the cell, and what's good for the cell isn't always what's good for the body. But there are cases where those mutations can be beneficial, and learning from those can also guide us towards new strategies and solutions to treating disease. Like what are our own cells doing in response to a diseased tissue can lead us towards pathways for new drug developments that may target the same pathways.
Brilliant, thank you.
For just moving to the last question, just say you all had in your seats feedback cards. Please write your views on it, and also to leave, if you'd like, your email to hear more and get involved in healthy aging research. And if you'd like to just give it at the end as you exit to me, and I'll collect them all. Promise. Okay, so yeah, for the last question, thank you.
Thanks. This was a brilliant talk. Early on, you drew a distinction between lifespan and health span. And I wondered, firstly, if you could say something about the variation across species in health span. Are there many species that have a significant lifespan beyond reproductive cessation? And secondly, about mechanisms, if you wanted to, you've been drawing this link between somatic mutation and cancer to explain the variation in lifespan. What else might you need to think about if you wanted to consider a variation in health span?
Yeah, so yeah, two good questions. So the first one, so yeah, we have much less good data on health span in animals. So for example, but we do think there are significant differences. So studies in birds suggest that compared to mammals, birds seem to kind of live healthily often and then just drop dead without often a decline in health. But how much of that is driven by the fact that we're much better at picking up signs of aging in a mammal compared to a bird? So it is hard to notice the signs of decline in health in many species. It really disentangles that. And it's true, during the talk I often talk about lifespan and aging, and they're not really the same thing. So lifespan is something that varies between species, whereas aging is something that happens within a species, a decline in function with age. But there are species that don't seem to show evidence of kind of aging or senescence. So even in the naked mole rat, it's been argued they don't really show an increased mortality risk as they get older. They just seem to age, and at some point they die of something. But it's not that their mortality risk doesn't increase the older they get. So there are species, there's this debate which species do and don't show that aging phenotype. And then, sorry, the second question was? Yeah. Yeah, that's a great question. So within somatic mutation, there's potential, we think, for example, in the blood system, that you get somatic mutations that change the cell fate trajectory, so you get a bias towards certain lineages of cells within the blood system caused by these somatic mutations, and that can lead to things we think like excess inflammation. So there's this idea that inflammaging is a big part of aging, this chronic inflammation. And so disruption of cellular function from these mutations we think could be contributing to that, for instance. But then there's many other aspects of decline in health with aging, like immune senescence, and it's really unclear whether or not somatic mutations play a role in these other aspects of aging. There's research to see if it does, but for sure there must be many aspects of this decline in health with age that are due to metabolic changes or immune system changes that probably are not to do with somatic aging. Again, it's just one of the aspects. For us, it's kind of like the metaphor of a car that breaks down over time. You've got to kind of keep all the systems working. Mutations are one of those systems, but if you keep the engine running, then your tires can still wear out. So we kind of got to fix all of these pieces, which is why there's not really an easy solution to the whole aging issue. But we think the genetic changes could be one of the important parts of the system.
That's marvelous. Well, thank you very much, everyone, for joining us tonight. And thank you, Alex, for such an enlightening, spectacular talk. If anyone want to ask some follow-up questions, feel free to take advantage that we have Alex with us. Have a wonderful evening. Thank you for joining us.
There you go.
So quick, man. Thank you.